like to do in this last talk is convince you we're at a major tipping point where I think we can really think in a finite time of transforming medicine in a fundamental way. And some of you may know about one of the first major tipping points in medicine, which occurred in 1905 when the Carnegie Institute had a report on medical schools and hospitals. And these were all independent operations. There was no real teaching. It was mentoring kinds of things. And the question was, where should medicine go in the future? And the report came to two conclusions. One was that science should be brought into clinical practice, a radical idea at that time. And the second was that medical students should be taught science with the clinics and so forth. And one medical school really leaped to that opportunity, Hopkins. And over a period of 30 years or so, it really propelled itself to the very top position in medicine where it's held today. And again, I think the tipping point we're looking at today has to do with the fact that scientific wellness is absolutely going to transform the practice of healthcare. And that's mostly what we'll talk about in this lecture. But I'd also like to really stress the importance of systems medicine, because it's a complicated entity that not many people understand. And a big part of it is the new technologies that it has introduced that are dramatic. And I'm going to talk about a fair number of them, not in detail, but to give you the picture of what these are going to be able to do for us in the future. So to get started, let me say, I remember when I went to Caltech in 1970, being a little staggered by the complexity of disease and frankly of biology. And at that time, we didn't have systems biology, which was a way of formally thinking about complexity, nor did at that time we have a lot of the technologies that we can use to think in very different ways about what to do for medicine. So I started early in my career at Caltech and was involved in a series of paradigm changes for the next 40 years, actually, that all led to, one, dealing with complexity in various ways, but two, to putting a focus on what was going to be essential to medicine in the future. And the first of these paradigm changes was to develop a series of 6 instruments that could read and write DNA. And bringing engineering to biology had two important consequences. It introduced the idea of generating lots and lots of information, high-throughput biology, and it led to the idea of big data in databases that could be used to analyze various aspects of medicine. One of the instruments, the automated DNA sequencer, got me invited to the first meeting ever on the Human Genome Project. And immediately I was really captivated with that paradigm change, because it was utterly clear that it was essential to have a complete genome, to be able to think in a global, holistic systems manner about human experiments and human disease. In fact, from developing the DNA sequencer, that led to an opportunity in cross-disciplinary biology. That is, the realization that leading-edge biology should always be embedded in a cross-disciplinary context where it could drive the development of relevant technologies and of analytic tools. And I tried and failed to persuade Caltech this was a good idea. But Bill Gates made it possible to come to the University of Washington and set up the first cross-disciplinary department, which was quite successful. But it became clear to me that a state university had real constraints, and the sense of administrative bureaucracies for doing things that were truly new. And that's why in 2000, I resigned and started the Institute for Systems Biology with Alan Derriman and Rudy Abersol. And of course, the vision here was to apply systems thinking to both biology and medicine. And one of the first things that led to was the formulation of a series of concepts about medicine, systems medicine on the one hand, and a conviction that healthcare should be predictive, preventive, personalized, and participatory. And we called that P4 healthcare. And the idea then was, how could we bring this P4 healthcare to the healthcare system? And in 2014, we actually set up the first experiment on what came to be called scientific wellness, where we used dense dynamic personal data clouds, repeatedly gathering information across the course of a year with 108 individuals to follow wellness and actually to optimize wellness. We'll talk more about that. And then, of course, the final paradigm change came, as you heard, last year when ISB affiliated with the Providence St. Joseph's health system. And our mission was to become their research arm, and I to become the chief scientific officer. And this gave us direct opportunities, as you heard earlier from Tom, to really think about bringing systems thinking to medicine. So what exactly has systems biology contributed to thinking about systems medicine? I would say 4 things that are really important. One is the idea of these dense dynamic data clouds. The second is the idea of dynamical network biology that can be used to understand biology and to think about diagnostic and drug targets and so forth. The third are all the technologies that we'll spend some time talking about. And the fourth, I think, are very focused ways of looking at data. So with regard to the first, dense dynamic data clouds, the idea here is we can gather this data. It's dense because it's billions of data points. It's dynamic because we collect blood in the first study every 3 months. And of course, it's personal because we focus on each individual in this context. And what we were able to demonstrate is if you integrate and analyze these data, you end up pointing toward actionable possibilities that let one either improve their wellness and or avoid disease. And we'll talk in some detail about that later on. Now, this is absolutely the personification of what precision medicine should be. And one of the challenges, of course, is it's expensive because of the cost of the assays. And we'll get to that a little bit later. The second idea is, of course, that the information highways of living organisms are their biological networks that operate at many different hierarchical levels, and that these networks specify development, physiology, and aging. And if disease-perturbed, they cause disease, then if you can understand how those disease-perturbed networks differ from their normal counterparts, you get new insights into diagnostics and therapeutics. And we'll talk about some interesting examples of those kind of things. So what about technologies? I would say this kind of thinking had been somewhat defined in 2006 or 2007. But what was very clear at that time is, although we had a whole series of technologies we were interested in, we really couldn't get NIH to fund most of them because they were too wild or speculative or there wasn't enough data. And I had the very good fortune to, at that point, meet the minister of Finance of the state of Luxembourg, who had decided to transform Luxembourg's economy from a 90% dependence on financial services to bring in healthcare and biotech. He asked ISB to write a proposal to help them do so, and we did a bunch of things for them, which I won't talk about. What he did for us was he gave us roughly $100 million over a period of 5 years, 2008 to 2013, and we used that to develop of the order of 10 systems-driven technologies and strategies and so forth. So let's talk then about some of those things. I'm going to give you vignettes of a lot of these in the future, so I'll only talk about the orange ones at this point in time. Rob Moritz, together with Rudy Abersol, really pioneered probably the most important contribution in mass spectrometry for protein analysis, something called targeted proteomics. And what Rob went on to do was to create a library of all of the peptides you needed to be able to analyze by targeted proteomics or SMR proteomics the entire spectrum of human proteins. And he put that in a database called the SMR database and everything. So that really is an important tool that we'll talk about later. So technologies are straightforward kinds of things. What we mean by systems-driven strategies are things that are more complicated, where you need both a computational framework to put together different bits and pieces, and of course, family genome sequencing, which in 2010 we showed was an incredibly powerful way for identifying disease genes within a family. Gustavo Glussman and Jared Roach did that. But more recently, Gustavo, with his colleagues, have done some beautiful new methods for computationally comparing thousands of genomes. And if you're interested, you can talk with him. And a really important part of systems contributions are to use a knowledge of dynamic networks to think about drugs. And Nitin Baliga gave absolutely a gorgeous talk on exactly how we do that. But let's go and talk about big data and how we view that. So one, you always want to have the data be as global or comprehensive as possible. You never know what's important. Number two, it is really important to study dynamical data. Dynamical from a temporal point of view or from a spatial point of view. And that is one of the ways for really dealing effectively with signal-to-noise problems. Number three, integration of data gives you new insights into how biology operates and so forth. And number four, this idea that there are theoretical concepts like dynamic systems approaches that take enormous complexity and compress it into very simple concepts, I think is really going to be important for the future. And we're starting to see repeated examples of where this has been done. And in the case, actually, of Jim Heath, he's actually developed some really beautiful things using thermodynamic phase transitions and biology to take very complicated data structures and make simple hypotheses from them. Creating the platforms for the systems-driven kind of technologies is key. And what is also really key, and it's something that IT companies and a lot of big biotech companies really have trouble understanding, is that in all big data sets, there's relatively little signal for any biological question you want to ask, and there's an enormous amount of noise. And the noise comes from two places. One, it comes from technical limitations of the data generation technology, but even more important, it comes from other biologies that are relevant to the biology that you're looking at. So you have to learn how to subtract those out, and we'll show you an example of that in just a moment. So I'm going to run through and give you very rapid vignettes of a whole series of systems-driven technologies and strategies at this point of view. And of course, one is the idea we can use animal models where we can induce disease, follow it all the way through the progression to the end as a way of looking at the true dynamics of a disease. And prion-induced neurodegeneration was the first of these, where we took inbred strains of mice, injected into the brain's active prions, and this disease progressed over a period of about 22 weeks. And we generated brain transcriptomes, roughly 10 of them across that time period. And in each case, we subtracted the diseased animals' transcriptome from their normal litter mates. So we only looked at things that differed. And to give you an idea of the noise, almost 40%, 30% of the mouse genes changed in the mouse brain. And it turns out after you create a series of specially constructed inbred strains to get rid of the noise, and I'd be glad to tell anybody who's really interested how to do that, it turned out that we had really 300 genes that mapped into this disease process. And we could use those genes then to identify 4 major networks that were a part of this whole process. And what was really striking about the first observations we made is those 4 major networks became disease-perturbed in a temporal order that was always exactly the same. Prion accumulation first, followed by glial activation, innate immune responses, and then 2 forms of neurodegeneration. And the key here, and it's the key for human disease, is if we know what the first disease-perturbed network is like, can we reverse the disease at that earliest stage before it's ever manifested itself as phenotype? And you can see prion accumulation becomes perturbed many, many weeks before the phenotype ever shows up at all. But one of the real questions you can ask yourself is, you can't sample brains to look at neurodegeneration in humans, so what are you going to do? And of course, the answer to that is there's supposed to be a brain here. I hope it doesn't cancel out too many other slides. Anyway, that's a brain, and there are of the order of 100 or so brain-specific proteins that map into a whole series of brain functional networks. And these are proteins that actually can be detected in the blood, so brain-specific blood proteins. And what we were able to do for the mouse is to find 11 of those proteins that mapped into various of those 4 networks, and to demonstrate by mass spectroscopy, targeted mass spectroscopy, that the activation in the blood was seen only a few weeks delayed from the activation in the brain. So you could follow the initiation, and you could follow the progression of the disease in the blood. And in fact, we'll talk now about other organ-specific blood proteins. And this is really a key technology for a new platform that we're starting to think about, where blood becomes a window into looking at the dynamics of human biology and human disease. So what we've done is analyzed about 20 databases for transcriptomes and proteomes, and we've identified for 19 different organs a whole series of organ-specific kind of transcripts that you can see here. And of course, once you have the transcripts, the real question is, how many of those transcripts actually can be secreted or shed into the blood in a manner that they can be detected by mass spectroscopy. And it turns out that for some organs, they're very easy to find. The liver, we have enormous numbers. But for the brain, where we have almost 700 brain-specific transcripts, the number of brain-specific blood proteins that we've discovered is strikingly low, of the order of 20 or so. But what you can see here for a whole series of organ-specific blood proteins are the fact that they differ across 6 orders of magnitude in the blood. We need all the sensitivity of mass spectrometry. But the other question we must ask ourselves is how many of the proteins we don't see are present in the blood, but they're just below the limits of what we can detect. And there are new technologies coming that will allow us to answer that kind of question. So we now have, we've looked at, identified 1,600 transcripts in 19 major organs, and we've looked at about half of them and created the SRM peptides we need to identify them in the blood. So we have of the order of 200 some organ-specific proteins from 19 organs, and the missing ones are either low levels or not there. And of course, for the brain, it's really interesting to see, are they kept in there because of the blood-brain barrier? So we're beginning to look for them in individuals that have diseased brain where the barrier has released its constraints. We don't know the answer to that question. We've used them to study and get markers for liver fibrosis, liver toxicity, and Lyme disease. And the Lyme disease studies were really cool because we looked at a person first that got acute, eight people actually, that got acute Lyme disease, and then we looked later for the organ-specific proteins, all of them we had. What this enables you to do is identify all the organs that become pathologically modified by the process. So in Lyme disease, there were 5 organs that were changed and 2 that were really strikingly changed. So one was the heart and the second was the liver, interestingly enough. We're using these as one view of blood as a window into looking at dynamics. And our goal in the future is to be able to get a microfluidic chip that can analyze of the order of 1,000 or so proteins, say 10 to 20 from each of 50 major organs in the body. And what that becomes is a wellness chip. And we can envision doing that at home with a cell phone that pricks your finger, takes a drop of blood, does the analyses, and ships it off for analysis. So that is the wellness assay of the future. Third-generation DNA sequencing is really going to transform what we can do. And this is a generalization. There are many different approaches out there, and I'll show you a really cool one in just a second. But the ease of sample preparation is incredible. Today, it's a nightmare to do sample preparation, and it's really rather expensive. It's single molecule, it's nanopore, it's electronic detection, and you're going to be able to detect all 16 chemical modifications of DNA, the epigenetic marks. In principle, for some of the things, you can sequence RNA every bit as well as you can sequence DNA. And of course, the enormous parallelization is going to lead to being able to do the genome in 15 minutes for much less than $100. And about a week ago, I saw an incredible new approach to nanopores, and it's the first time I've ever seen it, it uses capacitance rather than ion current to detect the changes. And capacitance has 2 big advantages. One, its changes are measured by electrons rather than ions, and it means this device is going to be 10,000 times faster than any of the conventional things like Oxford nanopore sequencing that we have today. And number 2, it has an enormously increased sensitivity. So it will easily be able to detect the 4 bases and the modifications that we talked about later. Okay, how about using, again, targeted mass spectrometry to do biomarker discovery? And these are collaborations we did with Integrated Diagnostics, a company we started 5 or 6 years ago. And the first problem we worked on was we see of the order of 3 million, or pulmonary oncologists see of the order of 3 million nodules in the lung a year in the US. And about 600,000 of those go to surgery, and about half or more of those are surgeries on benign nodules. So the question is, can we create a diagnostic that could distinguish benign from neoplastic nodules? And the approach we took was to identify almost 400 assays for proteins we chose in various ways, but they were all supposed to be blood proteins. Interestingly enough, we could only detect about half of those proteins, but we used those then to look at the plasma from 72 individuals with diagnosed cancer nodules and 72 with benign nodules from 4 different sites, and doing it from multiple sites is absolutely critical to dealing with human polymorphisms. And then we used a really clever systems algorithm which said, which are the proteins that are most successful in interacting with one another in a diagnostic panel? So we took the top 32 proteins from the 190 that we'd scored, again, depending on how accurately they made the 144 calls. We took the 32 and we randomly scrambled them into 1 million panels of 10. We scored the rank of all the panels, and then we identified 13 proteins that actually were very cooperative, found most frequently in those panels that were the best for diagnostics. And then we did a validation study and got even better score with one new site. And we actually, Integrated Diagnostics was able to commercialize this panel. And the reason they could commercialize the panel was that it had the ability to identify 36% of the benign nodules, which was really very good at the time. But now we've used other systems approaches to go from 13 to 2, and we have a panel that can now identify more than 50% of the nodules. And if you calculate what that'll save you in terms of unnecessary surgeries, it's north of $4 billion a year. So that was the real driver for getting this thing done. So we've used that for that approach for lung nodules, and then together with Seroprognostics, who did the work, we did it for preterm birth, and again, we got a binary panel that was superb at 19 weeks for detecting women who would go on to preterm birth so that you could give them progesterone, delaying the pregnancy and getting a normal birth. And we've done this with PTSD, glioblastoma, liver disease. I won't really talk about those. And a really pioneering technology that Jim Heath didn't talk about when he was here are peptide protein capture agents. And the basic idea is you make a library of about 10 to the 6 circular D-amino acid peptides randomly generated in sequence, and you use the protein that you'd like to make the capture agents against to identify monomers that have low affinity. Then you can use a chemistry called click chemistry to join 2 of those monomers together in the appropriate 3-dimensional orientation to create a bimer or a dimer or a trimer or even a tetramer. And with each mer, you get of the order of a log increase in specificity. And the really key point here is that those circular D-amino acid peptides are rock stable and everything. So what this means is, one, you can send them to Africa in an envelope, and that specificity will never go away. Number two, they are as sensitive as you want to make them. The dimers have specificity, sensitivity that's approximately that of some of the best monoclonals. And what is really cool about this is once you know, say, the two circular peptides plus the structure of the linking reagent, you can synthesize them in vitro with copper catalysis. So you have the opportunity to make very large quantities of things. And what you can do here now, Jim has figured out how to create these peptide reagents against peptides, and that cuts down the cross-reactivity that plagues antibodies in setting up ELISA assays for complex mixtures such as blood. So we think we really can reduce the cross-reactivity. And what Jim has shown is that we now have techniques where we'll be able to produce these in very large scale. So you can do in vitro or in vivo diagnostics, and more recently, he's demonstrated in several cases, these are going to be terrific drugs that can be very precisely targeted to exactly where you want them pointed. For example, the mutation in KRAS, he targeted perfectly and showed that capture agent almost didn't recognize the wild type RAS at all. So my prediction is these will replace monoclonal antibodies within the next 10 to 15 years. So if you want to make a lot of money, you ought to invest in this company soon. Anyway, single-cell analyses, and this is a case where we looked at the development of iPS cells to cardiomyocytes. And it was a beautiful collaboration with Sui Wang and Callie and Rishi and Barton in my laboratory. And again, this is a beautiful example of how complex theoretical biology can give you deep insights into things. So the simple analogy is we know now if you take a whole population of people and you analyze them in a pool, you're going to miss all of the essential features of the individual members of that population. And the same is true of an organ. If you analyze all the cells in the organ, you miss the very essential features of the individual cells in the component. And that's part of the justification for doing single-cell analyses. And what Callie and Rishi were able to show very, very nicely is in the process of differentiating from an iPS cell to a cardiomyocyte, there were a whole series of points at which simple binary decisions had to be made. But by analyzing the transcriptomes, you could make predictions long before that decision was actually officially made as to where it was likely for most of the cells to go, either to one differentiated form or to another differentiated form. So it simplified all the complications of looking at tens of thousands or thousands of transcripts anyway to simple kind of binary decisions. And that's what single-cell analysis is going to do in many, many different dimensions. And the final technology I'll talk about has really been pioneered by Mike Tyers at Montreal. And this is an absolutely incredible technology. And the big idea is, how can you synthesize 1 billion natural products really rapidly? And of course, a related kind of idea is, how can you screen them really rapidly for the drug targets that you'd really like to see? And the strategy that Mike has selected is to take all the metabolic enzymes that exist in the gut microbiome. DNA synthesis is cheap, so you can synthesize them, and you can put them in cassettes that can be put together in different blocks to create different combinations of synthetic reactions in YAC vectors. And then you can assemble these randomly constructed YAC vectors into a library as big as you want to make it. And then you can carry out screening either at the level of looking at proteins or at the level of looking at cells. And what this gives you the ability to do then is literally to create billions in principle of these natural synthetic products. And the thing to remember is about 2/3 of the drugs that we have today come from natural synthetic products. And so you're really biasing the dimension of search space that you put these things into. And where this is going to be absolutely incredible are in things like drug resistance, where you could very quickly generate new drugs that would be able to interrogate completely resistant tuberculosis or things like that. And of course, it would be great in generating products for emerging drug and infectious organisms and so forth. One of my favorite quotes is this Freeman Dyson quote: "New directions in science are launched by new tools much more often than by new concepts. The effect of a concept-driven revolution is to explain old things in new ways. The effect of a tool-driven revolution is to discover new things that have yet to be explained." And I think that sums up a lot of the technologies that you've seen here. So in 2014, we really began to see a convergence of this big thing that you understand now as systems medicine, with the digital revolution that Mike talked about, big data and its analytics and social networks. And it allowed us to refine with considerably more detail just exactly what P4 medicine, P4 healthcare was about. And it differs really strikingly from contemporary medicine. It's proactive rather than reactive. It looks at the individual rather than at populations. It's all about including wellness as well as disease, and not just disease. And of course, it's really about using these dense dynamic personal data clouds to interrogate the integration of the environment and the genetics within the individual to look either at wellness or to look at disease. You can use it, as I'll show you, in either way. What is really interesting, I think, is it argues very much against the way we do clinical trials. 20,000 individuals, give them a drug or a placebo, extract the data, yet you know every individual is unique genetically and environmentally or lifestyle-wise. And that means you're averaging disparate objects, and when you do that, you enormously increase the noise over the signal, and Big Pharma spent billions of dollars demonstrating the truth of that. So what P4 medicine does is we take 20,000 individuals, each with their data clouds, and we stratify them according to the properties that we're interested in, the ability to respond or not respond to a drug. When you've done that, you can immediately identify the properties that distinguish those two populations. And just to drive that home, let me say, here are the top 10 selling drugs in the US today. And the orange individual is the one that reacts to the drug successfully, and the blue individuals are those that don't react. So the range is 1 in 4 to 1 in 25. Most of the drugs you give don't help the people. In fact, due to probably cross-reactivities, they probably harm the individual. They spend a lot of healthcare dollars. And when you're done with the regular clinical trial, say for this study, you have no idea how to detect this guy from the other guy, whereas if you look at them as individuals, that'll come out absolutely immediately. So the big idea then is P4 medicine is about wellness on the one hand, about disease on the other hand. And we know that probably 96, 97% of healthcare dollars go to disease today, and almost none to wellness, because there is nothing that people have done in terms of experiments and so forth. So we were thinking about how to be able to quantify wellness, how to be able to analyze it in a scientific manner. What I think is really important to point out is, again, the 4 different things the dense dynamic data clouds can do. So one is they can give you data that lets you optimize wellness. Number 2, or avoid disease. Number 2, they allow you to follow the initiation, progression, response to therapy, and clearing up of a disease. And number 3, and this is the most important one because it is the preventive medicine of the 21st century, they give you the ability, as you follow large numbers over a long period of time, to see wellness to disease transitions for all common diseases, and to use these systems approaches to reverse those diseases at the earliest stage before they ever manifest themselves as phenotype. And of course, we can use N-of-1 experiments too to deconvolute enormous complexities, and where I think this is going to really be applied very successfully first is in nutrition. And I think we're really in the dark ages with regard to nutrition at this point in time. So Nathan Price and I in 2014 thought that a pilot project on what we came to call scientific wellness, because it was quantifying wellness, was what we would like to do. And of course, there were 2 really compelling reasons for thinking this is really important. One is the idea that the population is aging. For example, 50% of the kids born in this calendar year will live to be 100. And the question is, what's the quality of their life for the last 20 or 30 years without scientific wellness? And number 2, if you look at the determinants, as a number of studies have, of health, you see that 30% genetics, 60% lifestyle environment, and only 10% environment. And of course, the dense dynamic data clouds let us integrate the combination of the 2 most important elements of this together. So what we did was to persuade 108 of our friends over the course of a year to be studied by these dense dynamic data clouds with a genome analysis to start with. And then every 3 months, we got blood for clinical chemistries and metabolites and proteins, every 3 months for gut microbiome. And then we did quantized self with the tracking devices and things like that. And that generated a database that, when integrated and analyzed, led to different actionable possibilities. And of course, we gathered the information over 3 3-month periods. And how we brought the actionable possibilities to the individual were through coaches who, 1, explained what the actionable possibility was to the individual, and 2, had spoken prior to every individual to determine what their real health objectives were, so the actionable possibilities could be framed in the context of their objectives. And this really worked well. 70% of the actionable possibilities were actually followed up and everything. And I'll give you just a few stories of what really happened. In the first blood draw, it was really striking. 91% of the individuals had nutritional abnormalities, 68 inflammation, most of it was dietary, 59 cardiovascular complications, and about the same diabetes. 53 of the 108 were pre-diabetic. We moved 8 to normal. We moved many of the rest of them over much more toward the normal phenotype and everything. Every single individual had multiple actionable possibilities and recommendations to follow. And of course, one of the most interesting ones, this illustrates 2 important things. One is the power of integrating different types of data together, and the other is how little we know about some things even today. So we had something like 90 of 108 pioneers were very low on vitamin D, and we immediately moved to give them all 1,000 international units of vitamin D, and for most, didn't even touch it at all. And what was really very interesting is we later discovered that there are 3 genes that have 6 variants that block vitamin D absorption. And if you have multiple of those variants, you need 10 to 15,000 international units of vitamin D to even move yourself up into the normal stage. Now, what was interesting about me is I had none of those blocking variants, but I needed 15,000 international units to get mine to budge up. So it means there's a lot of genetics we don't know. So you have to have the data here to be able to sort those out. And clearly a physician that only looked at the one dimension of clinical chemistries wouldn't have the insight to do this, because people only need 1,000 international units. It wouldn't be healthy to give them 15,000 international units. So, and there are a lot of reasons why low vitamin D is a bad thing to have. So if you ought to go check it and get it fixed if you're low. There are more than 300 genetic markers that predispose, give you warnings about potential athletic injuries. And for most of them, you can do proper exercises and so forth to avoid them. And what's also interesting is your genetics can optimize your exercise and can optimize your diet in really interesting ways. We had one individual come in with neuralgias for 3 years of all the major joints, and in the last year, a Bell's palsy, a facial paralysis of the 5th trigeminal nerve. And it turned out he had almost no blood vitamin B12, so he got intramuscular injections and completely reversed this process that had plagued him for years and years and years. Another example is 3 pioneers initially had very high mercury levels. And it turns out, again, not something you want to do, but 2 were due to eating tuna sushi. So you don't want to eat sushi from any fish high on the ladder of fish, swordfish, things like that. And 1 had amalgam fillings, and when they were replaced or when they substituted salmon for tuna sushi, the levels came back down to normal very nicely. And a final example was a friend of Nathan's Price at Illinois who lost his ability to concentrate and motivation, very high-powered physician. And the bottom line was he had almost no iron in his blood, and that replacement within weeks reversed the symptoms. So those are the kind of things you can deal with if you have the kind of information that one has generated here. Pioneers like the idea that given information, they can control their own health objectives in parallel with their physicians, presumably. That your genome doesn't control your destiny, just your potential. Many things we can reverse. And of course, I think what's most important of all, most of them discovered they weren't nearly as well as they thought. And in fact, if you think about wellness as this big ladder, I would say most of our pioneers were somewhere near the bottom here. And of course, the actionable possibilities move you up this ladder in an interesting way. And what we've discovered in integrating different types of data in the course of these analyses is you generate new actionable possibilities. So they will go on absolutely indefinitely into the future. And of course, the real idea is we'd like to get our pioneers up to Eric Topol's elderly. These are people that are 80 or 90 years old, never sick, never in the hospital, never taken a drug, and they go into their 90s healthy mentally and physically. And that's certainly what we aspire to with scientific wellness. And of course, that means scientific wellness is going to have to be a lifetime journey. And That's, I think, one of the fundamental revolutions that will be coming actually in healthcare. And even more than that, I think we'll follow individuals from fertilization all the way through to the end of life, continually optimizing, continually looking at early transitions and reversing them, and so forth. Here are the people that worked in the wellness project. You can see it was really a complicated kind of project here. And I've colored in orange here the individuals that really played a major role in helping us analyze data, which we're going to come and talk about in just a few moments. But the second thing about this group of people is at the end of 2014, pioneers really wanted to know where to go next. So we started a company in 2015, and we moved the heart of this group of people right into the company so it could get started in a very rapid manner. Airavail was the name of the company, and it was really interesting because from day one, you could begin to see most people aren't well. They come in with things that they know about beforehand. So we have an enormous repertoire at differing levels of things that we can follow in the context of disease back to wellness kinds of transitions, the inverse of what we talked about before. And the business plan now is we went from 100 to 1,000 in the first year or so of the operation of the company, an order of magnitude increase. And at the end of this year, we hope to be at 12,000, and we hope to go 300 up to even close to 1 million in 2020. And if people are curious, we can talk about just how we do that. I will say this company has the best CEO I've ever worked with in my career, and I started a lot of companies during that time. And again, these people played major roles in the data that we're going to talk about now. So the data from these studies, I look very much as I looked at the Hubble telescope, which could look at the heavens with a resolution heretofore unachievable. So dense dynamic data clouds give us a view of human biology and disease that is enormous in its dimensionality. And we have actually just published a paper in Nature, or it's in press in Nature Biotechnology now. And in that paper, I think there were 3 major kinds of observations, and I'll tell you about them, and you can see what they are as we go through them. So statistical correlations of the 6 different data bits in different data types, with statistical positive or negative correlations with data types and other types of data. So these are all the correlations between genome and clinical labs and microbiome and so forth. Number 2, we were able to demonstrate beautifully that we can determine genetic risk of any individual and correlate that with disease phenotypes in most interesting ways. And number 3, we saw all sorts of transitions between wellness to disease, disease back to wellness, wellness to greater wellness, and so forth. So in the statistical correlations, we could do 2 types of correlations. One would be all the pioneers at a given point in time, static analyses. And here we saw that there were about 4,000 statistically valid positive and negative correlations of a data bit in one type of data with data bits and other types of data. The second is we could look at correlations across the 3 different temporal periods of data gathering. And there we gathered about 2,000 data points. These aren't, the statistics aren't nearly as good there. But what is really striking to me is the 2,000 are quite different from the 4,000. So you're looking at different aspects of biology in really an interesting way. What we can do is take these statistical correlations and we can put them in a gigantic hairball. And then operationally, and this isn't exactly what we do, but it gives you the idea. What you can do is ask a program then to break the edges that have the lowest statistical probabilities in order. And when you do that kind of a program, what you see are discrete domains emerge out of this giant hairball. And what's interesting about the domains is they correlate with different aspects of physiology, they correlate with certain kinds of biomarkers, and they even correlate with various kinds of diseases. And we will take one of these communities, and we'll look at it in a lot more detail. So here is that cardiometabolic community here. And what you see is many, many compounds, proteins, and metabolites that essentially go in and interact in positive or negative ways with other things in the community. And we isolated one of these, protein called FGF21, and earlier studies in the literature had demonstrated it had the ability to reduce levels of triglyceride and to reduce levels of cholesterol. And remember, we found this in an entirely unsupervised kind of manner. And what turns out is it's a metabolic regulator that really does reduce these things, and it's now in clinical trials by Lilly. And of course, the interesting question is, there are many, many more things connected in with here, and they all have the potential of being biomarkers and/or diagnostics. How about determining genetic risk for the 60 or so gene diseases that are mapped by genome-wide association studies? And of course, what you do here is take the negative variance and the positive variance. They're all associated with probabilities, so you can create a statistical summing of these things within a single individual. And what we've also done is that for 2,000 supposedly normal genomes we have, and you get, more or less a bell-shaped division here. So this individual, if we looked at a number of different diseases, might be at very high risk for cardiovascular disease, might be at very low risk for diabetes or for Alzheimer's, or at intermediate kind of risk for breast cancer and so forth. So what you get is a whole profile, and we can look at the individuals and follow them with regard to their highest risks and looking for earliest transitions. In many cases, we know at least have some idea what those transitions might be. So for example, with this LDL example I talked about, we can bend the pioneers into 5 categories from very low risk to very high risk and intermediate risk and so forth. And what's really compelling is the LDL cholesterol levels correlate beautifully with the genetic risk. Okay, and we know in this particular example, if you use drugs to reduce levels of LDL cholesterol, you can improve cardiovascular risk. But the really important point is there are a whole series of other things, and I haven't labeled them here, that also correlate here. And what role would they play, and could they be biomarkers, or could they be even better targets than the LDL cholesterol is? And to give you an example, we did a similar kind of study in ulcerative colitis. And what the literature had demonstrated is that cysteine, which is a part of a synthetic pathway that leads to reduction power, and that's very important in dealing with cancer and inflammation and things like that. Cysteine correlates beautifully with the degree of ulcerative colitis activity. You see, that is, the more activity you have, the lower your cysteine gets, So really interesting question is, if you raise cysteine, will that help you with your disease? Now, the other thing that I didn't point out before in the correlations is that the things that correlate can correlate because of your genetic risk or because of your disease. And so the key question is, which is it? And in this case, it's utterly clear. The cysteine correlates beautifully with your genetic risk for ulcerative colitis. So it doesn't have to do with ulcerative colitis itself. But the other really interesting point is, can we change the genetic risk of these high individuals by giving them cysteine before they ever get the disease, and you don't allow them to move into a point where they transition? So we don't know. I mean, that's speculation. Here are a list of the GWAS diseases that we can do analyses on. And actually, the ones that you see here are diseases we're going to be doing clinical trials at Providence on, and I'll talk about one or two of those. So, transition states from wellness to disease. I have a good friend who's in the audience. I remember telling me a number of years ago about his family history of very bad cardiovascular disease, and several generations of fathers and grandfathers had died from it. And he followed his own LDL cholesterol, which was absolutely fine until he got into his mid-30s, and then it really shot up to be very high. And what he also noted is his weight at that same time really increased in spite of the fact his diet didn't change. And we know now that reducing LDL cholesterol can put you in good shape. But what this shows is, as you move into middle age, you begin to see these kinds of different disease transitions, and the dense dynamic personalized clouds will be ideal to look at these changes. In Aravale, they've seen now a whole series of different transitions from apparent wellness to disease, and it's just the beginning of many, many more we're going to see. And to give you an idea of the power of what this is about, we recently had one of the individuals diagnosed with stage 4 pancreatic cancer, which is a pretty grim diagnosis, unfortunately. But we had a whole series of earlier blood draws on the woman before the diagnosis. And the question was, in this very first one, a year plus away, could we detect anything that looked interesting with regard to a diagnostic marker? So what Aravale did was to analyze this earliest blood. They did it for almost 1,000 individuals, and they looked at a whole series of metabolites and proteins. And one particular protein was really interesting because it was an enormous outlier, and these are the rest of the individuals with that same protein level. And it's a notch signaling pathway. It's a regulator of cell growth. It's expressed in beta cells of the islet. And what is really interesting, now, I'm not saying that's a marker you got to do clinical trials, but it's really an exciting idea. And with 100,000 individuals, we'll be able to see this. But the really interesting idea is, suppose it was a great biomarker, and we saw this back here in this patient. What could we do to deal with pancreatic cancer? And I'd say there are two exciting things we could think about. One is we could activate immunotherapy to go in at a very low level and remove the tumor. Or two, we could do triple drug therapy for the cancer at, again, low levels, and see if, as with age, you could turn around that kind of possibility. And there are other possibilities as well. And of course, there are studies that suggest for many diseases, we'll be able to see things way, way before. So how we're going to deal with them, I think, is going to be absolutely fascinating. And we're looking at the other bloods now. So what you'd like to see, the best outcome, would be to see this thing increase as you go across the other bloods that there. So what we're looking at then with 10,000 or so individuals is the ability to see the early disease transitions and to develop systems approaches to be able to create biomarkers and eventually therapies to reverse before the individuals ever have the disease manifest. And this is preventive medicine, and it's one of the biggest items that's going to transform the cost of healthcare. State transitions and aging. This is very, very new and really exciting data. Approaches we've thought about are to follow individuals who go from less to greater wellness and look with regard to metabolites and proteins, and there are some interesting results there. But more recently, we can follow individuals as they age and just measure the degree of variability in range of various omic analyses. And we've seen that they really increase significantly, and I'll show you that in just a moment. We think we eventually will be able to do it for both physiology and psychology, so we'll have metrics, quantitative metrics of wellness. And they'll allow us to distinguish physiologic age from chronologic age. So if you're 78 and you're physiologically 25, you're in great shape. If the inverse is true, you're in pretty terrible shape. So I think these metrics are really going to be transformational for optimizing wellness and things like that. Here is the range of variability in the omics, starting with individuals that are 20 to 30, 30 to 40, 40 to 50, and you can see the median goes up just beautifully, and it's what you'd think about as the system gets older, you lose control. But what is really interesting is look at the envelope of variation within each of these, and what I think that means is there'll be individuals that'll be very, very low on this thing, and individuals that'll be very high, so we can make relative assessments of age and so forth. Providence St. Joseph's, as I said, is really transformational. We're going to be initiating a series of translational pillars I'll talk about. We are going to educate, bring P4 medicine to physicians, healthcare professionals, and to patients, and I won't talk about the education part today. Providence has 50 hospitals. It's present in 7 states, 7,500 physicians, sees 5 million patients per year. It has of the order of 30 million electronic medical records, and it's a system whose C-suite is absolutely in sync with having ISB as its research arm and this new world of scientific wellness. So the translational pillars are joint partnerships between ISB, Providence, Airvale, who will execute the dense dynamic data clouds, and a company called Multiscale that has the ability to extract difficult to deal with clinical databases and put anonymized patient records in the cloud. The pillars we're going to be looking at are 7, and I'll talk about 2 because we have the funding to get those started, and we're well along at this point in time. But again, the clinical trials are going to be systems-driven technologies and strategies and dense dynamic personalized data clouds, so they'll be different from any clinical trials ever done before. Providence has agreed to put 2,000 of its own employees into the Airvale Scientific Wellness Program for a period of 3 years, and to compare them with 2,000 that are treated as they were before. And this time period will allow us, 1, to quantify the improved health for the individuals, and 2, to begin estimating the magnitude of the economic savings that come from scientific wellness. And the second pillar that we're working on is an Alzheimer's pillar that's being led by Jared Roach. And here we're taking a systems approach to Alzheimer's, not looking at one feature or one drug or one strategy, but to put together a whole global approach to dealing with how can we optimize communication, synaptic communication within a brain that has shown the earliest stage of Alzheimer's. And what we'd like to be able to do within a 3 to 5 year period is, one, have diagnostics that quantify the onset of Alzheimer's, and two, that show with this approach we'll be able to reverse it back to normal. So 2 final slides before a summary. Today's major limitation for scientific wellness is clearly the cost of the assays. And of course, my prediction is that they'll decline from maybe $5,000 over a 10-year period to less than $100. And I think avatars that will amplify the ability of coaches to reach out to people, apps that could do much the same thing, and the digital devices that Mike talked about will allow us to measure simple and interesting, complex phenotypes. I think an incredibly interesting phenotype measurement I just saw a couple of weeks ago is there's an app that can capture from your computer 60,000 strokes, and they can make unerringly accurate predictions about neurodegenerative diseases, or normal. So scientific wellness, I think, is going to be covered by health insurance and government payers once we prove that it's going to make decrease the cost of the healthcare system and so forth. And this is what is going to be necessary to be able to be more inclusive in the future. And how we create the coaches that can deal with a different socioeconomic group, I think, will be an interesting challenge. And one of the really key things, I think, are churches. I think churches can really help this kind of thing spread to all socioeconomic groups. Okay, I got asked a question last night, why is this going to be cheaper? Mike disagreed with me. And I'm going to run through a whole bunch of reasons, and you can think about it. So optimizing wellness lets us avoid disease transitions. Identifying diseases and reversing them early is really absolutely critical. And identifying the transitions early and reversing them, again, is the road to prevention. Increasing inexpensive digital devices let us make more and more accurate kind of phenotypes. And of course, as we learn more and more about wellness, we won't need billions of data points. Maybe we can do it with 5,000 data points. We can focus down, and maybe we can make simple tricorder devices that can measure those, make those measurements at home and everything. Moore's law decline and the cost of wellness assays. Following disease closely for which an individual is at risk. The metrics for wellness, it'll tell us who to focus on. 20% of the GDP is healthcare, and 86% of that is in chronic disease, and we're going to move everything toward wellness and away from both acute and chronic, we hope. And the scale of diabetes, obesity, and things that I think we can deal with. If we can really deal with Alzheimer's, that's $260 billion a year just in that ticket alone. And following things over the entire life, I think is really incredibly important. And what's also important is to assess the value of the data, which on the one hand is going to allow us to transform how we understand wellness and optimize individual health. On the other hand, it's going to transform how the healthcare industry actually carries out its functions. So we are bringing P4 medicine to the healthcare system at Providence. The dense dynamic data clouds let us assess both environment and genetics and point them toward wellness and disease as well. We can show that wellness can be optimized through the acceptance and acting on actionable possibilities. And of course, being able to identify earliest transition points and reverse them will save lots of money. A whole new field of scientific wellness is going to emerge. I think that in 10 years we'll far exceed that of the disease industry or the current healthcare industry. I think the costs are going to go down dramatically, as we've discussed. Scientific wellness industry, transforming healthcare industries with the data and so forth. And of course, if we can really get the costs down, we can democratize healthcare in a way that's inconceivable before. So, the end.